Hi everyone. Today we're going to be talking about the second half of 16S data analysis and data handling. So we're working on our workflow from 16S, pre-processing and data analysis, and now we're going to be handling the data to make it more suitable for our downstream analysis. What we're left with now is our taxa table from previously our ASV, OTU contingency, or feature count table, all meaning the same thing, and as well as our metadata table. So our metadata is something that we're going to generate ourselves, and I'll provide an example, but basically it's just data about your samples, whatever you think is meaningful or important to add as a detail. Our taxa table from last time is going to be that data about which taxa is assigned to which ASV or OTU. And then our ASV OTU table is going to be about how many counts of each ASV were found in each sample, right? So it'll be looking at the counts. This is kind of what they look like if you were to compare them. So our taxa table here has our OTUs in the first column, and then all of the taxonomic classifications of all the OTUs going down. Notice that each of these tables are interconnected in some way, and they all come together to create something that can be called a fileseek object, which is kind of a folder that can store all of the files and all of the tables into one object, bioinformatic object. This is more applicable when you're doing this through coding. But so we have our taxa table, and then we also have our OTU table or ASV table, and notice that they're going to be connected here by the first column. So whatever you have in the first columns here should be the same. And then we have our metadata on the right, and here in this case, the names of each of the samples or the sample IDs need to perfectly match, as well as, again, the OTUs need to perfectly match for the taxa table so they can all merge together or align or be compared. And so we want to make sure that there's no extra characters or letters or white spaces in our in our names here for each of the samples. And some examples of things you can put in your metadata are, your mouse IDs. For example, this is actually my metadata table from a previous experiment. We have our dose, so the dose of treatment that the mice received, what day, what sex the mice are, the age, genotype, and so on. And so these are all variables that later on you can test. So I always recommend including as much information as you can in your metadata table for whatever experiment you end up doing. Because you never know what you'd like to compare in the end, right? You might find differences that you didn't expect. Another thing I want to mention here is that data is sparse in the OTU table, and this is very normal. What I mean by sparse is all the zeros that we have. We're going to discuss that later and how that is potentially problematic down the line. Okay, so problem number one we're going to talk about is that sparsity. And that arises out of various biological and technical factors, but it's mostly because there are many rare or environmentally specific taxa only found, let's say, or only detected in certain samples and not in others. So if they're only detected in a couple samples, they're still going to show up um, as an OTU or ASV, but then they're going to be zeros for the majority of samples except for maybe a few columns. So there's going to be a lot of zeros that we find emerging, as well as potential PCR errors, contaminants, or sequencing artifacts that can also be driving these rare OTUs that aren't found in many samples. And then also, just to give you an idea of how many zeros you can find, I mean, up to 90% of OTUs or ASV tables can be filled with zeros, and that's not uncommon, right? Some ways to kind of address this is by filtering them out and removing any ASVs or OTUs based on a couple of principles here. So prevalence filtering will drop any taxa or features, you can say, that appear in too few samples. And here's kind of an example of what I'm talking about. So we have four samples with various different proportions of microbes. And so if we look to see it, how many samples do we find those red microbes in? We find them in all of the samples here in this case. And then the same with the green, same with the purple. But then in those curly yellow samples, we only have them in 75%, so 3 out of 4. But the blue microbes right here are only found in 1 out of 4 samples. And so that represents, let's say, this is a simplified example, but 25% prevalence. And so if we apply a 30% filter, that's going to be removed, right? Because it's not present or prevalent in at least 30% of the samples. Abundance filtering is a little bit different. That will drop features or taxa with low total counts. So if we were to sum all of the counts of a taxa across all the samples, there could be a minimum that we want to apply. For example, here we have a set of 4 samples, and then if we were to count all of the red microbes, let's say, we're going to get 15. Same with the green, we'd get 19. If we want to make a cutoff of 5 as our sum, there's a couple that wouldn't meet the cut. So here, these yellow curly microbes, there's only 4 of them across all of our samples. And so their total abundance would be 4, as well as the blue one right here. There's only 2 across all of our samples. And if we were to apply this 5 read filter, those both would be removed. Hopefully that makes sense. Okay. So how do we apply this? How do we know how much to use? It's really going to be a balance between sensitivity and noise reduction, because ultimately what we're doing is reducing that noise. However, we still want to be somewhat sensitive to potential rare taxas if that's important to our research question. So how do we choose our filter thresholds? Well, there's some things to keep in mind that prevalence and abundance filtering helps us with. So it can help us improve reproducibility because it's reducing that noise of potentially rare taxa. But this is assuming that rare taxa are or not prevalent taxa are not as important, right? But what this can also help us with is that it reduces data dimensionality. So this reduces the amount of multiple testing hypotheses if you're calculating p-values or p-adjusted values, and increasing statistical power. What does this mean? Well, if we have less OTUs that we're running statistical tests on, we have less of a chance of having false positives and therefore overall higher p-adjusted values. Right here. Another thing that we can look into is how it can stabilize any distances or dissimilarity measurements or ordinations. So it can kind of stabilize some measurements down the line, which we'll talk about in another time. The diversity or richness, the number of taxa will change as a result of removing some of the OTUs or ASVs, right? So filtering them out will reduce the overall richness of our microbiome samples. And this can, of course, alter then our results, our findings, and potentially distort them. But filtering also assumes, like I was mentioning before, the rare taxa carry negligible information, and that may or may not be important in your experiment. So that's just something to keep in mind. And then this is just one example of a filter that you could use is just over 10 reads as a sum, and then let's say 10% prevalence. Another type of filtering that we can apply is called variance filtering. This is optional, but it's a little bit interesting because it will remove taxa that are not really changing across samples. So taxa that are really stable, their abundances are pretty similar across all the samples, because this assumes that if a taxa isn't really changing, if it's constant, its abundance is constant across different variables or different samples, it's unlikely to be associated with an outcome or be driving or having an effect. However, again, this is context dependent. In your experiment, it might matter if something is constant or not. And this is relevant when you have an underlying hypothesis. So if you know of a particular microbe or of some kinds of microbes that you expect to have a change, you want to see what is changing rather than what is staying constant, that would be a place where you want to apply this variance filtering, for example. Some types of variance filtering you can apply are based off of the standard deviation or the interquartile range or this coefficient of variance. So if you're looking at the standard deviation to do this variance filtering, any low standard deviation OTUs will have low fluctuation, and this takes into account outliers, but this is one way to apply that variance filtering. Things that don't vary will be removed. So another metric is this interquartile range you can apply to, which is looking at the 25% to 75% percentile abundance. And so things that have very little variation will be removed compared to things that have a lot of variation. And then a coefficient of variance is another metric where the ratio of the standard deviation to the mean now will normalize the variance. The next problem we need to address is unevenness. So this arises from a different number of reads per sample that's detected through sequencing. So this means like an uneven sampling effort. That's another way you can say that, or sequencing depth, right? This also arises from biological or technical causes. So deeper sequencing is going to appear to give you a higher diversity because you're able to detect lots more different microbes. And that's going to give the appearance of more diversity, more different types of microbes, more rare taxa, simply because you looked more into them. A way to address this is called rarefaction. This is controversial, but still widely used, but it's used in specific ways. So you can't just apply this, and that's kind of a general rule for all these types of data adjustment techniques, is that everything is really context dependent. It requires kind of a justification for. In this case, so rarefaction is a procedure to subsample from each sample's read count to uniform length. Okay, what does that mean? This is also occurring randomly. So let's say that you have reads here on the y-axis, and each of these lines are a sample. And so, for example, this sample right here has a lot of reads detected, a lot of sequencing depth, but the one right next to it down here has very low sequencing depth, very low number of reads that were ended up being sequenced and brought to us in our OTU tables. So one thing we could do is that these look like, you can think of these as French fries or spaghetti that you can adjust. What we're going to do is actually cut them to the same length, okay? But do we want to cut them all the way down to the lowest one? That's a question you'd have to ask yourself, because let's say we rarefied them all the way down to this length, okay? Because we don't want to remove all of our data, right? So one thing we can do is choose, we can choose where we want to rarefy down to and remove any samples that won't fit that cutoff, basically, that won't make that cutoff. And so we'll rarefy down to this amount and then make all of our sequencing depth even in that way. This is a random process again. So what will happen is that if this turns out to be, let's say, right here, 1,000 reads that they're all rarefied to, there's going to be a random sampling. So it's going to randomly pull out 1,000 samples. Let's say this is 5,000. 1,000, well, those will randomly um, now appear here. Okay. And like I mentioned, anything under your defined rarefaction threshold will be discarded. So you're going to have to balance between, do I want more reads or do I need more samples if they're going to be discarded? And that's a balance you're going to have to make. And all samples will end with the equal total count that you set as your threshold for rarefaction. This means that you're giving all of your samples a leveled playing field in terms of sequencing depth. And we can observe this here closely too, because you might question, okay, how do I know what to rarefy down to? How do I choose the threshold of the number of reads per sample? Okay, this is something that can give us some indication. On the y-axis, we have richness, which is basically the number of ASVs found. And then we also have on the x-axis library size, which means the number of reads or sequencing depth of the sample. So let's say we wanted to see how the depth of our sequencing relates to the richness. So how many microbes or how many different kinds of microbes are we detecting overall? And what you're going to notice is that at a certain point, the more depth of sequencing that you have, it doesn't really help in terms of increased richness. It actually plateaus at a certain point. And that's going to look different from all your samples, but here I would say it's about here where it starts to plateau. So let's say that you do have a sample that only has 1,000 reads. It actually ends right there. Okay. That might actually be okay where um, you could rarefy right here, right? You could rarefy all of them down to about 1,000 or so. And that won't necessarily change your richness, but that will change many, many, many other metrics in microbiome analysis. So it's really important that we're only applying rarefaction for certain things, for example, richness, which is part of alpha diversity. So that's one way you can decide where to set your rarefaction threshold. So there are the critiques, right? The first one being is that we're throwing away valuable data, ultimately reducing our power. So this is really important for different types of analyses. And another criticism is that it's kind of arbitrary, your choice of rarefaction, but we try to make our informed decisions based off of our rarefaction curve. But there still is going to be bias in the end. A key thing here, one of the types of microbiome analyses we should never use rarefaction for is differential abundance, because this could result in false positives. Let me show you how. Let's say we have two jars, identical number of marbles, red and blue, in each jar. Okay, let's say we want to remove 5 out of them randomly. Okay, because of random chance, they might not be the same colors of marbles that are getting removed. So we might have more red marbles removed in jar 1 and blue in jar 2 by random chance. What we end up with is two jars that are no longer identical in their proportion um, of, um, of the marbles, or the abundance of the colors of the marbles. So what that might look like is something like this. Even though jar 1 and jar 2 had identical blue, red, and blue marbles, now it appears from random chance that the abundances have changed simply due to that random removal. So now it appears that jar 2 has more red and less blue. Okay. And that's also argued in the waste not, want not paper as your reading material. So this remains common for diversity metrics, but that differential abundance is not recommended to be used. Another problem we face in microbiome research is compositionality. So this has been discussed previously, but basically all taxa abundances are measured relative to your sample's total read count. So scale is lost. That's something I want to emphasize is through the bench workflow, through the sample collection and DNA extraction and PCR and sequencing, we no longer have the true scale of the amount of microbes there were originally, but rather simply a proportion, a ratio of all the microbes. The implications for that is that it's hard to tell um, what is really driving the changes in the ratios of our microbes. This example here is a common one. This is one example where it appears in this relative abundance. So if we're looking at the proportions or percentages of the microbes changing, it looks like Bacteroidetes decreased in relative abundance and Firmicutes increased their abundance in ratio. But there's two things that could have driven that, or more than two things. But here are two examples, which is in scenario one. In scenario one, we have Bacteroidetes actually decreasing and Firmicutes staying the same. So in this case, it drives making the percentages 25 to 75. In another situation, we might have both Bacteroidetes and Firmicutes increasing, but let's say at different proportions or different rates. So in the end, Bacteroidetes is still 25% of Firmicutes. Another problem this brings is one of scale. So you might have a situation where the absolute counts, and it's not impossible to get an idea of the absolute abundance, but it requires different assays, different experiments to do. But as far as our 16S microbiome data, here's another distortion or limitation. So we're not fully aware of the scale that was originally in our sample in terms of absolute count. So we might have a sample that has 70 green, 500 red, 100 yellow microbes. And then another sample that has exactly the same ratio, yet 10 times more of all of the microbes. In the end, we wouldn't be able to tell, right? So in the end, no difference would be detected after sequencing because at the end of the day, we're just taking a scoopful out of these cups, these theoretical cups. We're taking a scoopful, a subsample, and then normalizing them throughout the process. And that subsetting happened at extraction, at PCR, at library prep to go into sequencing. There are some ways that we can account for this. We cannot take this feature out. It's inherent to the process of microbiome data that true scale is lost. But we can apply some normalization techniques that account for this. So normalizations allow us to make reads with differing sequence depth, more comparable, and normalization can also rescale as well. So here are some of the techniques. We have total sum scaling, cumulative sum scaling, upper quartile normalization techniques. This total sum scaling, briefly, it's just able to turn those counts into percentages, and this will be helpful for that relative abundance calculation. The cumulative sum scaling, upper quartile normalizations, will normalize using the lower 75% of data. This is assuming that most abundant taxa are driving the largest difference in the total number of reads. And then this is used especially when few taxa are dominating. Something critical to keep in mind here, though, is not to pre-normalize if you're using a tool, a count-based tool that already has normalization built in, because you don't want to double normalize. And so therefore, you should be using the normalization techniques only for analyses that don't need those raw counts. Some more ways we can normalize are the relative log expression, the trimmed mean of M values, as well as CLR transformation. Okay, so each of these are actually built into tools, bioinformatic tools, but just to go through them, The relative log expression is able to estimate how much to scale for each sample, and it does this through a median of ratios, applying this into the DSeq2 tool, a very widely used tool. The trimmed means of M values is able to trim those extreme taxa and then create a weighted average log-fold change. So this is inherent to the Edge R tool. That's also widely used. But I want to focus our attention to the CLR transformation, the centered log ratio. Okay. So this centered log ratio has this formula right here, where the CLR with subset I represents the transformed abundance for one feature in one sample. I here represents a single taxon, so taxon 1. X represents the counts in sample 1. And then G represents the geometric mean of all the taxa in sample 1. This ln is the natural log. And so what we're looking at here is putting the count of a single taxa in one sample in context with the rest of the abundances of the other taxa in our sample 1. What is the geometric mean, though? So in particular, the geometric mean is different than the arithmetic mean that we're used to calculate averages, which is by adding and then dividing by the total number of numbers that we have. So if we were to add all these numbers up, we would get 2,000 and get the average 2,540, which is quite far from most of the other numbers because it's being skewed by another number. This happens a lot in microbiome data where extremes are able to pull the average. So we use the geometric mean, which instead it actually multiplies each of the numbers and then takes the square root of how many there were. What we get in result is 150. In this case, it's actually finding an average that's much more closer and less pulled by the extreme values. Okay. Another thing to keep in mind about the natural log that we have right here is that let's see, we cannot take the log of 0. And so this is going to be problematic if you remember in our OTU table right here, we're going to face many, many zeros. So what we need to do instead is apply something called a pseudo count by replacing all these zeros by 1. You might wonder, is this going to distort the data? And the answer is yes, it can change the data. However, it is required for this CLR transformation to take place. What the CLR transformation is able to do ultimately is show how much higher or lower this taxon is compared to the sample's geometric mean. So the average, the geometric average of the abundances of the other taxa, how does this one compare? This is used for, and these are down the line, but PCA plots, correlation plots, clusterings, and this is going to be internal to the Aldex 2 tool. In summary, just to look at all of the different types of handling techniques. So we have our phyloseq object, which will apply the filtering to prevalence, abundance, low variance, normalizations, which will help improve the evenness and distortions of our data and the variance of our data and rescaling of our data, such as rarefaction. And then we also have transformations, which is mostly CLR. One really key thing to keep in mind here is that these steps completely depend on your data. They depend on your assumptions in your experiments. They depend on the type of downstream analyses that you wanted to do, diversity, abundances, and these steps can significantly alter the representations of your findings. They can significantly alter your findings. And so what's required is a biological and statistical justification for each of these steps. Let us know if you have any questions, but that's all for now. Thank you so much.